Great, there's also that. So, hello everybody, and thank you very much for coming to the antibody validation for immunohistochemistry session. We are kicking off this session with our first speaker, that is Jelena Luic from Cornell University. Jelena obtained her PhD from the University of Novi Sad in Serbia. After a postdoc of St. István University in Hungary, Jelena moved to the US in 2020 to work in the laboratory of Paula Cohen. She is studying contraception in human and mouse models. Jelena is a new member of the ICAROS consortium, and she aims to validate antibodies on male and female germ cells.
Thank you, Fabio, for introduction, and also thank you for inviting me to participate in this session. And I have to say, this is my first time being in this conference, and I'm really amazed how well organized it and how many amazing people are there. So. Thank you for having me. Today, I'm excited to tell you more about ICAROS platform for those who are not familiar with it. First of all, just brief introduction to add to what Fabio said is I'm coming from Cornell Reproductive Science Center, so I'm not a member of any core facility. I'm a researcher in the research lab of Dr. Paula Cohen. She is a well-known reproductive biologist who is studying for decades reproduction, mostly focused on meiotic division and events happening especially in prophase 1 around crossing over events. Also DNA repair pathways happening also during meiotic division and being one of the crucial aspects of producing functional and healthy gametes. Also gene regulation during whole spermatogenesis and oogenesis process, but we like to say that the sexiest and the most exciting project in our lab is about developing novel non-hormonal male and female contraception strategies. Since we are experts in meiosis, our approach to contraception is very unique. So we are studying, we are aiming to target early meiotic events for contraception purposes, contrary to many other labs who are working on this aspect of the contraception. They're targeting like post-meiotic phases, sperm production or sperm function or fusion events between sperm and egg. We actually wanted to start much earlier and to prevent any leaking of possible spermatozoa because only one spermatozoa is necessary for fertilization. So we wanted to start much earlier in spermatogenesis. But we also have our own strategies for developing non-hormonal female contraception, also targeting mid-meiosis. Before joining Paula's lab, I was also a reproductive biologist from my PhD time in Serbia. I continued to work in reproductive biology during my postdoc in Hungary. In this period, although I was a reproductive biologist, my work was mostly focused on applying some very modern biotechnological approaches for supporting different fish population on site. So we were kind of emergency team who worked on conservation programs for different fish species all around Europe. And also another part of my work was devoted to ecotoxicological studies, where my job with my team was to assess a lot of different fish tissues through histological assays, which we use as a biomarker of effects of different cyanobacteria and cyanotoxins from aquatic ecosystems. So during my whole career, I was exposed to a lot of histology, I have to say, but just when I moved to Cornell, I actually realized the importance of good and reliable and robust antibody systems, which we actually use on a daily basis when we study meiosis, because meiosis can be studied mostly through chromosome spreads, and when you make chromosome spreads, you want to label all proteins involved in meiotic events with specific proteins, and those have to be very specific because a lot of events are there just puncture, like small dots, so you want nice, clean, and specific antibodies. All of us here, we absolutely know what is the purpose of antibodies, but neither of those uses of antibodies doesn't matter if that antibody does not recognize the protein of interest and the target protein. So from that point of view, I just want to emphasize again that specificity and selectivity of antibodies are the most important aspects of the antibody reagent, together with reproducibility and tissue context for every antibody which we use, so to enable good research practice and publishing. we all, I guess, realize that we are in big antibody liability crisis, and working on this presentation, I actually find out some very interesting numbers. One of those is that currently there are about 1.6 million antibodies out there targeting different human proteins from 200 suppliers. Here we have to take care about the information that actually a lot of suppliers just reselling antibodies, but under different serial numbers. So we have a lot of antibodies who are actually the same antibodies but sold from different vendors under different catalog numbers. And also here I would like to say that those 1.6 million antibodies are actually not covering all human proteins, of course, for different reasons. And there is a huge problem, which actually very important for us in contraception studies, that the huge part of human not genome, proteome, is actually in a section of so-called dark proteome, for which we actually don't have antibodies developed for different reasons. So those proteins are actually understudied or not studied at all because we don't have reliable tools which we can use for those studies. This is very important for us because when you want to work on developing novel non-hormonal strategies, you definitely want to target certain proteins, but you cannot study those proteins because you cannot study them if you don't have certain antibody targeting that protein. But those proteins are also not of huge interest for companies because those are just specific for spermatozoa, and companies don't have a huge like financial interest in developing those antibodies because they are very, very narrow niche and very small market for those. So we are meeting those issues regarding these numbers as well. Recent studies from actually Icarus Group show that more than 50% of all those antibodies out there at the market are unreliable and actually don't perform as they're advertised. Talking about finances, I just learned that from one of our company partners that they spent just in last year $2.6 billion just for developing and validating antibodies for neuroscience research. And another side of the coin are research laboratories who, according to some old data, but I guess it's just even worse today, waste up to $2 billion per year in total on bad or underperforming antibodies. So this is huge waste of money, time, effort. Our grant money and resources are already very limited, so you definitely don't want to use your PhD students or your technicians to waste time to optimize antibodies which actually don't work very well. And this problem was emphasized the last time, although there were like a lot of alerts during the time and in the last 15 years, I would say. Maybe some of you already saw this article. It was published in November 2024, where actually the author mentioned Icarus platform as one of the possible solutions for this crisis. So today I will talk to you about Icarus platform. Icarus stands for Antibody Characterization through Open Science Principles. This platform was established about 8 years ago by this amazing group of people from McGill University in Canada, led by Al Edwards and Peter McPherson. The whole group is a group of neurosciences and neuro researchers. So they were firstly interested in characterization antibodies for their own research and different neurodegenerative diseases. And then they very quickly recognized that there is a huge interest from companies from another side in collaboration on this type of effort. So they decided to set very high and very ambitious goal to characterize antibodies for all 20,000 human proteins. Although we know that it will be very, very challenging, they start building their platform on very solid grounds, setting a few key criteria on which they will proceed during the time with this project. First of all, they're industry independent and there is no bias in antibodies which are used in their research or any comments on their reports regarding the like favorizing any antibodies. To enable this, they actually build a huge consortium, and it's still ongoing process, so it's continuous process. We are adding new companies every year. Right now, they have 13 antibody-producing companies on board who actually made agreement with the Icarus platform to provide all antibodies and necessary reagents in kind for outsourced characterization by the Icarus team. In order to systematically organize their efforts, they actually decided to develop a strategy which will be disease and topic-specific target groups, so they can acquire financial support from different granting agencies or nonprofit organizations depending on their interest, disease interest or topic interest. For example, we are funded by Male Contraceptive Initiative and Bill and Melinda Gates Foundation for our contraceptive research and our antibody characterization for contraception research. Both sides of the consortium, together with the Icarus team, agreed to open science principles, and it is one of the key criteria on which all participants have to agree. So all results obtained from this research are absolutely open science, cannot be used for any commercial downstream work, and all reports are published through Zenodo platform, F1000 research platform, as well, where you can find the full reports for each protein target, protein target, and all validated or characterized antibodies for that protein. Through Zenodo platform, you can actually access all high-quality images for each of those characterized antibodies. And as I said, there is a very strict policy that there is no conclusions, recommendations, or anything like that. So they just show results, and it is on researcher, whoever is reading the report, to make their own conclusion and according to their preferences to choose antibodies which they want. I will just show you very quickly how one of the reports look like. I just picked random. I don't know anything about Huntington disease. So this is F1000 research portal, which is, as I said, open science, completely available for everybody. It is also open for peer reviews. Anybody can do review. We always encourage people to do a review, especially if it is their expertise, just to help us understand better the whole context of the protein target. And each report has very uniform structure. First aspect, because they decided to go with cell line, testing everything on cell lines, so they use human cell line database like DeepMap, where they actually choose cell lines which satisfy certain criteria of gene expression of the target, gene target protein. Once when they select cell lines, if there are more, they just use few of them to run very quick Western blot to see what is in which cell line they have the highest abundance of the target protein, so they proceed with that cell line. The most important thing in this part is that they always have wild-type knockout cell line as absolutely reliable positive control and absolutely reliable negative control, which is, you will agree as well, one of the most important aspects when we characterize antibodies. Knockout cell lines are usually provided by our company partners, so they are fully validated as well. Second step is antibody collection. Once when Icarus team or any company or any funding agency nominate the target protein, we send a request to all our antibody manufacturer partners to send us their antibodies. We won't start characterization process if we don't have, let's say, 2 or 3 antibodies available at the market. And we usually favorize characterizing renewable antibodies because it is something which we absolutely believe that we have to support. So recombinant and monoclonal antibodies for sure. But we start the whole characterization pipeline only in a moment when we have all antibodies in-house, because one of the main principles when we come to the characterization process itself is that we run all antibodies on the same material in the same time under exactly the same condition. So each report has 3 three main figures. First figure is about characterization antibodies for Western blot. Very simple criteria for failed and passed validation process. Figure always looks like this, depending how many antibodies we get from the company partners. It looks like this. Each column is one antibody. For each antibody, we have wild type and knockout protein extract. As I said, it was run side by side, same conditions. The only thing which we respected from manufacturers is their recommended concentration of antibodies. Everything else, we just use according to our standard protocols. Also important point that all antibodies which are provided from our partners, no matter of which method they recommend in their catalog, we run all three methods, which I will show you right now. So we can maybe discover something new about their antibodies, which also they didn't have chance to realize during their screening. And they're very grateful for that. So we have very good feedback. I will show you some numbers. Second figure is always about time on immunoprecipitation, also very simple criteria, and also figures look like this. As I said, there is no conclusion, no recommendations, nothing. We just give like very quick like recommendation about failing and passing the test, but nothing else, that's it. Order of antibodies is also random or alphabetical order, whatever we agreed with company partners for the certain target proteins. And the third image is about immunofluorescence. Since they're studying everything on a cell lines, they make mosaic, they plate mosaic cells with together wild type and knockouts in ratio 1:1. They stain them before plating so they can differentiate them later during imaging process. And you can see on the top left, for example, first two figures are from one antibody, so they always image on channel to acquire the antibody staining and also DAPI staining. They do some computational analysis to segment individual cells, and then they compare signals from wild type and knockout cells. So that's very briefly how the reports look like, and they always look like this. And one thing which is definitely missing from our characterization pipeline is immunohistochemistry. Obviously, they started with something which is the most common need, but also with something for which they were easily able to agree on protocols. But for IHC, they are discussing for more than a year with our company partners about like protocol which we will use for characterization. I think that this year is the year when we will start characterizing our first antibodies for IHC. But how we are thinking about this? I know that there are like different thoughts and different strategies on characterizing antibodies for IHC, especially because of very high requirements for this type of analysis. But our idea, it is still not a final decision, but I think it will be something like this. So they will take multi-step screening process where they will start with cell pellet screening step firstly, because they already had their cell lines, knockout and wild type cell lines. But they won't do just basic screening. They will do proximity ligation assay, where for those who are not familiar, they will just use two different primary antibodies that recognize two epitopes of the same protein or different epitopes from different proteins, which actually when you add a secondary antibody which is coupled with plus and minus oligonucleotides, if they are in close proximity to each other, less than 40 nanometers, they will actually hybridize, forming that circular DNA molecule, which will then be amplified by DNA polymerase. And once when you add fluorescent probe, we will get a signal which can be imaged under the regular fluorescent scope. With this method, we will, I think, try to overcome the problem with human material for which we won't have negative controls in the future, and all antibodies that pass this cell pellet screening step will move to histological sections. We are still debating should we use microarray tissue collections or regular tissue sections. And in this step, I think we will start including mouse tissues in the whole Icarus characterization platform, because so far they only cared about human proteins. But when they invited us from Cornell to join their ecosystem, we actually said, yeah, that's great, but we cannot move directly to human proteins when we talk about contraception. We firstly have to study them on rodent models. So we need antibodies characterized for rodent models. So I think that is one of the reasons when they recognized actually that characterizing antibodies for rodent model is beneficial. Also companies, when I presented to them our idea, they liked it as long as, they said, as long as we see financial interest in that and a good market, we will support you and we will send you additional vials of antibodies to test on mouse tissues. But also another advantage for using mouse tissues is that possibility of having transgenic mice and suitable negative controls. So the whole Icarus platform, I said, for this about 8 years of existence, built a very nice ecosystem of right now 13 antibody manufacturers. So far we have 2 companies that supply us with knockout cell lines, and through this ecosystem we have access to more than 85% of renewable antibodies currently available at the market. So far they characterized about 1,100 antibodies for about 100 target proteins, and from our results, more than 60% of those antibodies failed for one or more application methods. We have more than 100 publications in our open science portals. We attracted a lot of attention from the research community, but more important is that we attracted a lot of attention from our company partners, where they actually adjusted their catalogs according to results of our reports. So far they flagged about 400 antibodies in their catalogs, 73 antibodies they deleted from there and excluded from their catalogs, and 138 antibodies so far have been modified in their catalogs by adding or deleting some of the application methods. So we are very proud on our success so far. And I would say the cherry on the top of the last year was our publication in Nature Protocols, where together with the Icarus team, a lot of co-authors are actually from antibody manufacturer companies. So they're very tightly connected with the Icarus team, and they're working together with us on developing all these reports. So I encourage all of you to reach out and read this paper, because we explain the protocols for all 3 platforms in very details. But there are also some other publications in eLife also where you can find more details about the protocols which Icarus platform use. About strategic expansion, for sure, because everything started at McGill University at Neuro Institute. Biologists realized that actually they cannot characterize antibodies for reproduction. So they were like, okay, but if we want to characterize antibodies for all human proteins, we have to include experts in their fields. So, so far they are expanded their research community to Leicester University in UK.
We start discussion, sorry.
Sorry?
More 1 minute?
Yeah.
Perfect.
Yeah.
Thank you.
So far they have Leicester University from UK for cardio and pulmonary targets. They have Cornell University for reproductive biology and contraceptive targets. And then they have University of British Columbia for cell surface and cell death neurological proteins. For sure, we are looking for more partners and more sites which can help us to cover more human proteins with their expertise. So I encourage you all to engage with Icarus, test your antibody reagents, following Icarus consensus open protocols, open for yourself some new grant opportunities by including antibody characterization in your future proposals, join our review pool and review our F1000 reports, and become Icarus champion, and then help us expand to new sites. I would like to thank everybody here. I would like to thank Bill and Melinda Gates Foundation and Male Contraceptive Initiative for recognizing the importance of antibody characterization program, so they are invested a lot of funds for us to test antibodies for contraception purposes. I also would like to thank my lab and also to thank my colleagues from McGill University who also initiated the whole platform.
Thank you so much. And we can take some questions. So, any question from the audience about this project? Yes, please.
Please use the...
There is the microphone there in the middle of the corridor. No, the other side. Okay.
Hi, awesome work. I love this. I was wondering, what is the long-term goal for your database as a whole? Like, it sounded like you are mostly focused on human proteins, but are you considering at some point expanding that into other species?
Yes, so because I did it. No? No, it's good. Sorry. Yeah, definitely. So it was, I have to say, very hard discussion for me to convince Icarus people, but also to convince companies that characterizing on mice models is something which is beneficial for everybody. So I think we will move forward just with mice model for now. I'm not sure because how robust the whole platform is and how high requirements they have for result quality and everything, and also very limited funds so far. I'm not sure that we will expand to other species, at least for next few years. So human and mice, I think if we can manage certain, like a number of proteins covering with this platform, it would be great. But thank you for the question.
Okay, last question.
Hello. So this is great work. I am really excited to check out that website, but I have how searchable is the site? Because when I'm going through trying to buy antibodies, I want to see a picture and protocol.
So our reports are searchable on F1000 platform if you type protein of interest. Usually it's the easiest way, but you can always go to Icarus website, and on the Icarus website there is always a link to each protein which they already made a report. For now, they only have reports for neurosciences because they started from there. But stay tuned, there will be some reproductive targets as well for contraception soon. Through Icarus website, you can reach everything, but also we have Uniprot who is partnering with us. So very soon Uniprot will show reports from Icarus, all pictures, but also people from Leicester University from UK, they're working on amazing portal where they will make Icarus website just for reports and pictures. What you asked. But for us, you cannot imagine how many issues we had with company partners because they're like, oh, but which picture you will show first and how you will present this. And so there are like a lot of technical things, but since we have pretty good collaboration with them, we are discussing very openly all issues. So I think it will be out very soon during this year. So stay tuned.
Dad, are there any sort of consortiums, like say, I do all this work and I see a lot of the failures of this antibody. Can I submit those images somewhere so you guys can have a more realistic, like you guys do very good work, but is there any sort of consortium we could submit things to?
Thank you. Thank you for that. For now, we are just doing our internal experiments. So we are not pathologists. So that's something which, from that point of view, we cannot say. I can give my judgment about some reproductive targets and some pictures about reproductive targets, but I'm not sure that we will be in that situation very helpful because we work on target proteins, and when we get like funds for that protein or family of proteins, then we move from there. Sorry.
See, if you want to submit data, wait for the third talk.
Okay.
Oh, sorry.
Yeah.
Thank you. I'm Amanda Caney. I'm from Fred Hutch Cancer Center, and I'm helping co-organize this session. Thank you for attending, and it's my pleasure to introduce our next speaker, Zvi Makowski, from the Director of Microscopy and an instructor at the La Jolla Institute of Immunology down in San Diego. Well, in La Jolla, California, next to San Diego. Zvi got his PhD at University of Gießen in Germany, and then did his postdoctoral fellowship at La Jolla Institute for Immunology, where he stayed on and is now directing the core facility there. I wanted to share a couple of words that he had stated about what he does in his job, and he says he promotes honesty and openness in the best long-term research strategy. His greatest satisfaction is when his trainees return with improved protocols, better images, wiser analysis, and bolder ideas. And amen to that. That's fantastic.
So far, it worked pretty good for my personal statement in grants.
Yes. No,
Thank you very much.
Wonderful. Anyway, thank you, Zvi.
There's a QR code. I'm going to show it a couple of times during this talk. If you want to scan it, you're going to have access to the resources I mentioned here. So yes, there is 7 of us in microscopy and histology core, and we also work with Ken Kim, who is our histopathologist. Together we have a lot of experience, and we help a lot of different users from different institutions around San Diego and a couple of different places. So we also have a lot of perspective on the problems that people have working with different antibodies. And we make a lot of slides, and to make this, as a workable environment for our 3 brave sample preparation staff, Kasia, Natalia, and Simon, we want to make sure that they get this positive feeling when they are doing their work, and that is not turning into just waste of time and effort. So Sarah and Bill also are great additions to that team, and we, on the other hand, try to help with image analysis, microscopy, and development of methods. So this is not a biology or microscopy pit of despair. This is how we actually do research, right? You start with your biological question, you go through experimental planning, you do sample prep, you do image acquisition in our case, and then you do image analysis. And then you realize that you should better refine your experiment plan and improve your sample preparation and perfect your image acquisition, and then that image analysis is going to be a lot easier, and you just build in a little bit of reproducibility study into your own experimental workflow, and then you're ready to present the data. So there are lots of challenges with multiplexing, especially when you're working on FFP samples. We suffer from a lot of autofluorescence. There is a problem with cross-reactivity of reagents that we're going to be using that will result in something that looks like image on the right here, where those antibodies bound to the same target or were detected by the same secondary reagent. And microscopes in general don't have a whole lot of available channels. So I want to plug in the first plug for a video that you can watch from Quantitative Bioimaging Society. You can become a member for free and get access to that video library. Together with Sarah and Sripatram, we organize image analysis for multiplexing workshop at our place, and Sripatram, who works at Pfizer, presented that very nice talk about the approaches to multiplex. The 4S is sequential scanning, sequential staining, slide scanning, and stripping. And we like to do MUSIC because it has a better acronym, and that stands for multi-marker staining and image capture. So essentially, we have technology that allows us to stain for multiple markers at the same time and image them at the same time. His final conclusions and thoughts are that multiplexing can be merry because you get access to a lot of information, but only if your whole system is validated, and it can be a lot of mess if something goes wrong. So there's plenty of providers of technology that are offering great systems. They all have their pros and cons. Depending on what kind of system you're going to use, you're going to have to vary your validation efforts. In our case, we were doing a lot of that work in a traditional way, where we were using species and isotype-specific secondary antibodies. The problem is that even though those secondary reagents are cross-absorbed, they're not really 100% cross-absorbed. There is a paper that I listed here that does an amazing work looking into brains, and this is just beautiful stuff. Highly recommend to look at that if you're into neuroscience. We do a lot of work with the mouse tissues, and we find that a lot of good antibodies that are for FFP targets are made in rabbits. And because they don't have different isotypes, that's very limiting. So one of the traditional ways of dealing with that problem was to use HRP conjugated secondary reagents, tyramide, and then strip those primaries and secondaries. But when you do that, you need to be absolutely 100% sure that you completely removed the previous immunoglobulins from the run, because if that's not the case, you're going to have very weird results. And stripping can be harsh on tissue, and you can also get into that interesting problem where you've exhausted the spots on the tissue that can bind that tyramide. So order of addition of those antibodies is also important. Commonly used strategies to also use primary antibodies with haptens and then a secondary. A common example here is biotin and streptavidin, but manufacturers have invented a lot more ways to do that. If you use that, you do want to test and or block endogenous biotin, otherwise you're going to label mitochondria. Very popular DNA barcoded antibodies and the reporter strands, amazing possibilities to multiplex. The conjugation is complicated. They have shorter shelf life. Reporter can have some binding to the tissue, and directly labeled antibodies. All cheap, reliable chemistry, things that work in flow cytometry for years, long shelf life. Unfortunately, most of the microscopes are not sensitive enough to detect them. So here is where our platform is coming. We got it from a rare site. The microscope is called Orion. It has 20 channels. You do 18-plex panels. You stain all of your antibodies together, and you acquire them in a single scan. We use conjugated antibodies. No secondaries means less background. And the most important aspect for us is the freedom to choose the clone regardless of the host. We do single stain controls that builds an extraction matrix that we then use to resolve those overlapping spectra, and through computational processing that removes the bleed through between channels and also nicely unmixes out of fluorescence. The staining workflow is well fitted into how we were doing traditional multiplex in our lab. Go through the steps. And the only difference is that we try to reduce autofluorescence ahead of time by using LED and UV bleaching. Primaries go overnight at 4 degrees, and then the next day we would either probe with secondaries if necessary, or just stain for nuclei and cover slip. A cost is a very important problem for our labs, right? We, for quite some time, used this technology from Shandon that uses cover plates, and we spiked it up a little bit by building our 3D printed box. You can find design online. That allows you to use 100 microliters of an antibody solution to stain your slide. And to further reduce the cost, because Orion has such a big real estate on the slide, we will often put multiple pieces of tissue on the slide, and that really saves a lot of money for the labs. Now, we are all aware that we have the antibody problem, and sometimes you can find publications like this, and that's just a terrible news for the whole community. And sometimes you find publications like that, which are also news for the whole community because they have, and those efforts that you guys are making, right? This is an amazing work that helps us to save money and do our job better. But I also want to tell you that some antibodies can last forever. The fact that something has expired does not necessarily mean that it's gone. And you can use, apparently, some of them 26 years and more after the expiration date. So where do we find them? We rely quite heavily on CiteUp, which is a database of cited antibodies. And generally speaking, as a poor institute, we wouldn't buy, a license for something that would cost us money. So we very much like Nordic QC. That's more on the human side, more on the clinical side, validated protocols for looking at relevant proteins in the human. On this meeting, I learned about the EuroMapNet. I just heard about the Icarus. I checked them out before, but they didn't have IHC data. We look at Human Protein Atlas, and there are those two papers that I highlight here and here that really talk about how we can think about doing multiplexing validation. And I wanted specifically to highlight those two papers because they talk about antibodies that worked for them in their special techniques, but also antibodies that failed. And that saved us a lot of time when we were developing a panel for monkey tissue. When we buy antibodies, we buy them carrier-free, no BSA, no gelatin. We want recombinant antibodies or monoclonals over polyclonals because of the problem that those antibodies are not going to last, will change. And don't even get me started about the fact that we can still buy cell culture supernatants. That should be just forbidden. So our strategy for antibody optimization and conjugation is a fairly streamlined protocol. We will do antigen retrieval with both citrate and tris on two different slides. And we will hit those slides with 2 micrograms per milliliter of primary antibody and then apply secondary antibody. Okay, and we would put that secondary in far red channel that is the best for the microscope that we're using to study, where we have the least amount of autofluorescence, where the signal to noise is the greatest. And the first question that we ask is, has this antibody worked, yes or no? Do we have a pattern that we would predict from, going into the UniProt and checking where that protein should be expressed? Can we compare it with some published literature? Does it look like it worked? If it did, then we will analyze signal intensity. We will scan that tissue with 3 different exposure times, and that will give us an idea about the abundance. We will analyze those intensity signals. We will do assessment to whether that antibody is strong, and then we can put it into dimmer channel of our instrument, or if it's weaker, and then we need to pair it with a stronger channel on the instrument. We do conjugation via amine, and then we will again titrate that conjugated antibody to make sure that we see the same pattern as we were seeing before with the use of the secondary. So again, kind of trying to build that validation into the workflow. And you know what? Some antibodies don't like to be conjugated. If you kind of do a lot of shortcuts, you will spend a lot more time figuring it out because something will not make any sense. So the biggest problem that we always face is how to know what is actually real. And we should be more insistent on working with controls, but most labs cannot provide us with that. So what we ask them always for is like, give us positive and negative control tissues. Maybe you have some information from your other kinds of experimentation, flow cytometry, luciferates, gene expression. Give us something that we can grasp. If you have cell lines, make cell pellets, and those are very, very important resources. Maybe, we do no primary control, IgG controls. They're useful, but they don't really tell us about the performance of the antibody, just more about the performance of the system. And use knockout tissues as often as you can. This is from my graduate thesis, where we were expecting to see the staining exactly in those alveolar macrophages, exactly in that location in the membrane. It was even better in the knockout. And speaking of knockouts, not all knockdown cell lines are taking care of all of the isoforms. So, and also not all knockout animals are created equal, and sometimes you're going to have some weird stuff. You can do Sanger sequencing on those short constructs and validate that you know what you're working with. So some ideas for orthogonal validation. We do a lot of work with people that breathe and drink flow cytometry on a daily basis. This is a very rich resource for us. We can better understand, for example, how many cells we would be expecting in the tissue. But that's also a challenge that I'm going to talk about a little bit more in a different slide. I just wanted to highlight the ImGen, the database of immune gene expression in mouse tissues. And they are also coming up with ImGen maps, which is going to combine spatial transcriptomics data on a single cell level with some multiplex panels that we're also going to be helping with. So let's talk about false friends. If you would read that paragraph of text, which is taken from BioLegend website that talks about Syndecan1 being expressed through a variety of different cells, if you show it to immunologists, the only thing that they will see that this is also expressed on plasma cells, okay? And that's because they are used to that kind of gating strategy where they will drill down on that very small population and they forget to look for everything else. And that is definitely a false friend. And, IRF4, for example, would be a better antibody than CD138. So something that, we are exploring more these days is that those different tissues have very different levels of expression, something that I didn't really appreciate it before that much. Here we're looking at sodium potassium ATPase. It's titrated at the level where it's nicely detectable in the spleen. It's bright in the liver, and it's killing it in the kidney, right? We actually saturated the image here, the MFI, the mean fluorescence intensities for a larger region where we have a lot of dark pixels, so those numbers are not really representative. And just to give you a sense that we also get that kind of problem in our traditional IHC, where you're going to have those regions of tissue that are completely dark. Okay, so we definitely want to avoid that kind of situation. Saturation is your worst enemy when you're doing multiplexing. So some ideas for data sharing. There is this guideline for what kind of information we need to add to the metadata for highly multiplexed tissue images. And this is hard, okay? This is not trivial to do. It takes time, it takes effort. Hopefully, we're gonna be able to build that into the grants to acknowledge the extra amount of work that we need to do to do it right. And there are few repositories that allow you to upload those images that we generate. This is 75 gigabytes, and that's a relatively small piece of tissue. Doing that is also hard. It's very time-consuming. You have to provide a lot of metadata. It would be amazing if we could create tools that would allow us to take the metadata that we have and kind of transform them so, we can streamline uploading to things like Bioimage Archive. Another plug for a video from our 2023 From Samples to Knowledge CUPA training course, where Shripad Ran, again, who works at Pfizer and needs to validate targets for their clinical studies, shared with us how they do technical validation, not only of the staining, but also the whole assay, going from a 1,000 to 100 to 1 feed view, and kind of showing how, what is their approach for making sure that the whole system works and is nicely validated. So I would like to share some lessons learned and some interesting things that we kind of are starting to discover. I like the solution that we got because it allows us to cut down the cost. And this is very important because labs are struggling for money. And if we tell them, hey, we're going to gladly work with you, and we're just going to need to, for your final slides, we're going to generate an X additional slides so we can be pretty sure that this work is validated. And they'll be like, can you just stay in that one slide, please, for me? Thank you. So lowering down the cost is really, really important. And it allows us to try and test and expand our knowledge about how this works. The garbage in, garbage out is absolutely the case, and proper fixation is the number one requirement. And what we're kind of learning is that the tissue type for your final stain is super important. It can be wildly different, and sometimes also your sample preparation, small modification like inclusion of that UV bleaching step can interact with other components of your workflow and make a mess of your tissue. So, for example, we switched to citrate for mouse tissues instead of using Tris. So a couple of interesting observations. We had a human spleen slide sample that was sitting in the lab for about 11 years. And we hit it with a panel of our conjugated antibodies, and 5 of those surprisingly worked on that tissue, and those are looking promising. We would have to compare it with the fresh cut sample to validate that those targets are really making some sense and things like that. But there is also a bunch that didn't work in our hands. So an effort to kind of create that community resource for figuring out what worked in your hands and what didn't work in our hands would be amazing. And, we can all make mistakes, right? But I think if we will have a community vote of, 17, yes, this antibody worked in my hands, and 3, no, it didn't, that would be also very informative. So what can you do about validation when you work with unique samples that cannot be easily, reproduced, like those that are post-spatial transcriptomics runs? Well, you can connect to your peers and work together to establish center standards. And if you want to do that and learn how in our place, Miguel Reina Campos is leading our efforts to do multiplex IF after Xenium runs. Please reach out to him and get in contact, and we can talk more about the clones that worked. So putting it all together, those 2 papers are my starting point for that. I don't want to talk too much about this because it really depends on the system that you're using, but I would stress that you need to test individual components and the whole panel, and if possible, use the whole slide view and also zoom into subcellular locations. For things like edge artifact, where you can have a lot more staining with some antibodies at the edges, but other clones will not have that problem. And also trying to use the high resolution of our microscopes to see if that target makes sense. And I'm pretty sure that we're going to be having some surprises and some interesting discoveries along the way. So I'll stop here and be happy to answer any questions. Thank you very much.
I can get started. I'm kind of curious about, you're just talking about storage of metadata. Do you have an idea of what types of metadata we should be keeping and how we should be, utilizing that?
Yeah. So I can tell you that from my personal perspective, being a postdoc that started to do a lot of mouse work, I didn't have a system. And I was wasting a lot of time, trying to find the blogs, and keeping the information straight was very, very challenging for me. So now I created a system that I forced every user of the facility to use. But what I offer them is this kind of bind value proposition, where I tell them, look, people, you're going to have to keep those methods straight, and you're going to have to keep that data straight about your animals, your tissues, your treatments. Let's do that work once and do it well, and just recuperate that time every time on the next stages of your experiment. So what we are doing is we are essentially harvesting that metadata from the time when they put in work order for submission of their tissues. That works very well for traditional histology. That works very well for slow plexing, because we have enough spaces on our slides to kind of list all of those antibodies. And we actually have a problem with this high plex multiplexing, because it's kind of hard to put together that panel. So with help of Sarah, we developed a Band-Aid that essentially if you want to do some multiplexing with us, we're going to send you a Google spreadsheet. You're going to check off the clones that you need, and you're going to attach that to the submission in our core management portal, Infinity, and that's going to live together with those files, and then we're going to name this panel 73, let's say, and that's going to be attached as a part of metadata to the digital files.
And who's keeping that? Are you keeping that? Are you passing that on to the investigator? Does that travel along with their images for eternity? Like, who maintains it?
Yeah, so the research management plans that we have in our institution say that it's on the PI to keep track of this, but we're not going to be letting that data go. We're just going to store it in our internal database because I think it's going to be an important source of knowledge for us in the future. But this is one of the things that we haven't really solidified. We haven't figured it out completely, but we definitely see a need to sit down and think about it more carefully because it's important.
Are there any questions from the audience? Okay, we can move to our next and final speaker. Thank you.
Hello, everyone.
I was going to introduce you, Fabio.
Sorry.
So our next speaker is Fabio Sacchiarelli, pathologist at Weill Cornell Medical School. Fabio completed his residency at Sapienza University in Rome, Italy. He did a PhD in cancer proteomics at the Karolinska Institute in Stockholm, Sweden. Now he is still at the Weill Cornell Cornell School of Medicine in the lab of Dr. Massimo Loda. He works on prostate carcinoma there, but he has a dual function or dual role in that he also runs the MISI. It's a core lab for multiplex and high-plex IF on tissue specimens. So no further ado, thank you, Fabio.
Thank you so much, Amanda. So this is a presentation about a research group here in ABRF. We have been starting working on this, practically speaking, since last year, from September, October 2024. And here we are in the process of building this immunohistochemical atlas in the context of this association. First of all, I want to start with something that you probably know better than me, you know very well. So all the challenges that connect to the validation of antibodies when we are working with immunohistochemistry. And validating is a multi-approach task in which we face several problems, like validation principles that we often cannot apply for several reasons. Right models, right control. We discussed a lot about models with Elena and Zibiev, but not often is available, especially when we are studying a complex model like there is a human being, which you cannot manipulate the genome in vivo, of course. Problems of working with antibodies, you are searching for a reference validation point for immunohistochemical slides that often are not available. The best you can get is a small image, often not visible in a paper. And when you're working with so-called dark or exotic markers, this poses really a severe challenge since we don't know how to approach them. And the last one, we have this resource called the Human Protein Atlas from the group of Matthias Ulen, but I don't think this is really enough for our needs, as I will explain in 2 slides. This is a brief overview about antibody validation principles. It's taken from this paper from 2016. This is an overview of basically principles we should follow when performing antibody validation. So you can see the genetic model. There can be a small interfering RNA or a CRISPR. You can see the orthogonal validation, preferably work with an antibody-free protein quantification, like it be mass spectrometry, or it could be RNA, for example, in other laboratories. Independent antibodies, so 2 different antibodies for the same protein, recognizing 2 different epitopes. But of course, tagged protein also, so the overexpression of a protein in a mouse model or in a cell line, it can be also human, and immunoprecipitation, MS, that maybe is less suitable for immunohistochemistry for obvious reasons. So why we want to do a repository, why we are not satisfied completely with Human Protein Atlas? For several reasons. I think that the validation principles that apply there are not often stringent. There are no reproducibility data available in that atlas because the immunohistochemistry is performed just one time on that specific sample. There is a focus on only human. There is no interest into veterinary tissues. Majority of cases, only few markers, only few clones per marker are tested. Only immunohistochemistry chromogenic is available. Yeah, sure, it's the cheapest, but there are no other approaches available. And behind this atlas, this is a commercial purpose, of course, since Atlas Antibodies and Sigma are sending these antibodies. They're not for free. So what we want to achieve with this group? We want to create an online atlas that can depict basically immunohistochemistry antibody performance in a whole variety of markers, in a whole variety of tissues, in a whole variety of species. Why we want to be different? Because we want to make everything open source with raw data that are actually available for download. We want to have some reproducibility data available. We are interested in producing, until a certain point, some data redundancy across laboratories, especially when a batch of antibodies is available. And we want also to make sure we have an extensive metadata data set available for each immunohistochemistry there is a recording in the system, including the protocol that explains how that immunohistochemistry was produced. We want to be as much as possible rigorous. We want to set up a clear criteria for suitability of an antibody for that use, and also include a description of the performance. In the end, I don't think that antibodies can really be classified into bad and good, like black and white. Often is more important probably discussing and describing how an antibody performs in a certain settings, and then the user can make their own decision about the use instead of being black and white. We want to be as much as possible interested in multimodality. Immunohistochemistry chromogenic, of course, we'll include it, but there is also much more. Multiplex immunofluorescence, there is IMC. There are many other techniques available. And we want also to open for different animal species. So who are we that we want to do this initiative? We are all 32 members of ABRF. We come from 19 different institutions that are represented here. We have also some companies involved into this project. Our background is very variable. For example, me, I'm the pathologist, but we have several veterinary pathologists. We have many biologists, image processing analysts. We have many histotechs. We have many core directors. As you can guess, the majority of information, raw data that is coming from core labs, but not only. We also include immunohistochemistry performing research lab, and we also try to include some clinical data produced, so data were produced into clinical laboratories. For that, of course, we will need a proper IRB, and identify the samples, but we want also to include that. Our expertise in immunohistochemistry, multiplex imaging, and so on. For sure, we need a new good picture, because we don't have in our group. This is basically, very synthetically, the scheme for our atlas that we want to implement. We are just right at the beginning. I don't want to make this picture more complex, but basically what we want to do is submit in different immunohistochemistry or multiplex immunofluorescence raw data, plus metadata that are available, and we will go into the metadata in some slides. At this point, we want to have a data management system in place that can be, we're thinking of a Homero server, for example. And from there, the users, everyone is open source, so everyone can access it, will be able to visualize online the data, will may be able to do some quantification. We want also to include that in some versions, and also the possibility to download the raw data and metadata that are already available there. So, immunohistochemistry slides or multiplex immunofluorescence, any kind of imaging slide you want. We need the so-called good and bad antibodies. So antibodies you think are suitable for immunohistochemistry, but also antibodies you think perform terribly. Why not? We want to include also that. Whenever possible, we want you to include the batch number, because we want to make sure that we produce redundant data, and the data and the batch works maybe your hands, maybe another batch doesn't work. So in that case, we should discard the antibody, for example. example. We want to have the same antibody tested on the same tissue, as I said, in different laboratories. We want to have different approaches available in terms of modality. And whenever possible, open format, so files that are compatible with other formats. The list of metadata that should be included with the submission has been finalized. We're talking about around 65 different variables, okay? It's extensive. We are aware that it's big. We're totally aware of that, and I will show you an example of it. It includes information about the primary antibody, the secondary antibody, about the model you used. If it is human, if it is mouse, what kind of mouse model, as a cell line, what kind of cell line is that, how have you authenticated that, and also information about the processing, how many hours in formaldehyde, did you use Bowen, did you use formaldehyde, how did you process the sample, okay? This is a huge list of metadata, I'm aware of that, but we're trying to enhance as much as possible the reproducibility. And we want also to include into this metadata the protocol for the staining that has been used. I have also reported here RRID portal. This is something absolutely important we want to include. So a label that is unique for a specific clone, a specific catalog number, and a specific company. We want also to, of course, include the criteria that were used for the antibody validation. That is, it plays a very important role, since we need to make sure that the criteria we're using are reproducible and are clear. Here is just a list of, I know this kind of like an intimidate, I think, right? It's kind of long, no? It's sort of intimidating, but we think that the way for reproducibility, unfortunately, is this. We cannot take shortcuts. Here's the management system taken from the Omero website that basically, you can see that basically you can, through a client, acquire the data, put into the server of Omero, and then you can do your search, you can do search through the metadata, and do some analytics query, you can do some image analysis, and so on. What kind of output? As I said, I'm interested in having people visualizing their data, but also able to do a query across the data that we are building, to be able to download the data. In some cases, we want also to develop a sort of ICORO style report for each marker. So let's say I have 10 different antibodies for EGFR. I'm interested in building a report in which I summarize the results of the 10 different antibodies, so that maybe can help users to have a look at the data in a summarized way. And of course, one day we were interested also in offering some sort of data quantification for each tissue compartment. And where we are now, we are basically in the phase 0. So we want to build now a first prototype of data set on an Omero server. But then once the Omero server is up and running and it works, we will be ready for data intake from other contributors that can be core labs or research lab or clinical labs if done properly. And of course, we are interested also in one day into analyzing clinical data. So this is my presentation, and I would be really, let's say, grateful for you if you can provide any feedback on this project, honestly. Mm-hmm. We are just at the beginning, so thank you so much for being here first of all.
That's wonderful, Fabio. And
Thanks.
We are welcome to open the floor for questions for Fabio, for any of our speakers here. I have one thing actually
Yes.
for you, Fabio. It seems that, this antibody database is more, I mean, it's sort of like a crowdsourcing effort. It's taking our community to try to come together to provide resources. So how do we integrate data that's coming from other platforms, as we heard about earlier today, so that we, can make sure that we're not duplicating something that's also been done?
I mean, yeah, you mean when they submit the data? Mm-hmm. During the data submission, we are actually partially searching for duplication. Remember, we want to have some data redundancy. Mm-hmm. I think it's fundamental for reproducibility. So we are interested in having people submitting, let's say, the first same antibody and same tissue done in another lab. I'm interested into that.
Are we going to have ways to link to some of these other databases that are also?
Yes. I think we will be in the metadata for sure where the data were produced. It's already included into the data set, the laboratory in which the data were produced. I think we should for sure have some link, some way of connecting that to another web page and another thing. But I have thought of it, people are submitting, so I put the person submits and the institution and the core and the laboratory. So at least you can keep tracking to the laboratory.
I just want to say very quickly, I think that we have a big potential for collaboration because we are in the beginning of our IHC characterization. So let's see how we can manage to coordinate,
Absolutely.
to coordinate
Absolutely.
and actually help the whole community.
Yeah, absolutely.
Yeah.
Thank you so much. Yes.
Yeah, I think that this is an amazing idea.
Thanks.
For when you get to phase 2, I'm kind of wondering how you want to normalize the way that the data is potentially collected or submitted, so that way your analysis is actually comparable across labs. Like that, I think, is going to be a really, really big thing to potentially
Yes.
get labs all on board on. And I don't know, like maybe something like, Z's very standardized protocols that he uses, getting people on board to say, if you're going to submit this, you need to make sure that we're all following the same protocol, so that way we know it's actually comparable and that our analyses are also comparable across different labs.
This is absolutely true. Yes, this is absolutely true. For sure, you need to resubmit data, maybe make sure that people submit the data when performing a certain way and analyze only the data can be compared to each other, right? But it's also true that, let's say, some differences can be even appreciated when the protocol is slightly different. So, I mean, when you look for some antibodies in which we have basically a cross reaction somewhere, yeah, it's better if you do it in the same way with the same platform, with the same similar concentration. In some cases, the difference is so violently evident, let's say, they can say, guys, this antibody just doesn't work because in your platform performs like this, in mine performs like this. I see something completely different, you say this antibody is probably not reproducible. Or you are comparing two different clones for the same protein, and you know that from validation data you should not obtain that positivity in that compartment, for example.
I can see a use for both things being submitted, like what protocol that lab uses
Yeah.
for your stuff.
Yeah.
And then for like this kind of comparative analysis,
Yeah.
we have a standardized protocol that we use.
Yes.
We submit that.
Yeah. Yes, yes. It will probably require some production again of data. Absolutely, I agree with you. Yeah.
There are some interesting opportunities to, once that database exists, right? To also look at this from the image analysis perspective and find algorithms that are robust to those differences, right? And there is an evidence that those actually exist and we can use them.
Yeah.
And just before I forget, I'm sorry, the link that I posted is now open and you can all see it. And one thing that I forgot to mention during my talk is that each of us in this room is a knockout for about 20 proteins. And those are mostly the proteins that do not have a lot of known functions, right? So there might be this untapped opportunity, although I think it's still going to be cheaper and faster to CRISPR out something from a cell line than to find those volunteers, right, to provide their blood samples. But
Yeah, CRISPR and overexpression, I mean, they will save us, in part. Yes, please.
So I'm Angie from University of Colorado, and I just want to say I think this is great, and I know my boss joined the user group last year and is going to help to with the antibodies, but we are interested also in getting together with other folks at altitude, because we've struggled in some epitopes being at altitude, our antigen retrieval, so where we're going to standardize it for everybody, we won't be able to follow that because we boil at a lower temperature than everybody else.
that's amazing. I never thought about that.
It's challenging on our bonds, where most everybody will run their bond at, I think it's like 98 degrees or 99.
Yeah.
We can't go above 90, 93 or 95. I'd have to look, because if not, it bubbles.
You're working with Bond Rex.
Bond Rex.
Well, maybe
this
if you,
defect.
yes, you would use, a pressure cooker essentially, right?
That's what we do. We use the pressure cooker. We can't use PT module because we asphyxiate everybody on the floor because it boils off.
For, I think $5,000 you can get a very good one with a precise control of the temperature of the timing.
We do that as well.
There are, I think BioCare is selling that. BioCare.
That's
Yeah, there is a fantastic model, super precise. That is the top, seriously.
But I think that brings up a good point in that, this is going to be a very nice resource for our community to have, but we're going to recognize that all of our labs are going to run things a little bit differently. And so we will have some variations. And so that's where the metadata is so important to have in there. So I think, this will provide a good reference point, some, images that we know, conditions where it has worked in the past in that particular research institution. There's going to be fine tuning, regardless. But I think this is going to save us a lot of steps in the process.
Okay, we know that we have to add an extra 2 tablespoons of flour when we make cakes.
Yeah.
Yes, exactly. Yeah, unfortunately, you cannot taste a food on a website. Otherwise, it would be amazing. But no, you cannot. You can see immunohistochemistry. Okay, thank you so much. If there aren't other questions, I think we can close the session, right?
Yes.
It's time to.
Oh,
Oh, yes.
So, I think you know that already. I think this is great, but from a practical sense, this is a lot of work.
Yeah, it is.
So what is the planned structure for moving forward so that a year from now, maybe we'll be done with phase 0 or in phase 1?
We're in phase 0.
Are you envisioning like monthly meetings online, or like what
Yes.
are the practical benchmarks for ensuring that we move forward from this meeting?
Yes, so in 2 weeks, I want to call a meeting, 3 weeks, let's say. And during this meeting, we do, for the last time, we review our metadata to make sure nothing is missing. Then we will try finally to build our first metadata list, okay? So I will ask any contributor who wants to participate to add, let's say, at least 10 antibodies. I want to create a small data set. It has to be a pilot at the beginning, and add everything, okay, diverse. I don't know, it can be a GFP, it can be a Vimentin on a hamster, it can be everything you want. We need to test how our metadata are able to represent what we are doing, okay? This is important, that we're not forgetting something. Once we have that, we are sure that more or less our list of metadata is comprehensive. At this point, we need to start to collect the different formats. Okay, now someone is using Apereo, someone is using Vectra Polaris, someone is using, a Nomitif format, right? It will be a babel of different formats. For that part there, we need to have already a small version of a Homero server already available. Because in that case, you want to start to see what happens if you start to upload the images. You want to make sure that the repository can handle the different kind of data that will be there. It doesn't have to be that much. It can be 10 images, 20 images. But you want to make sure that it's able to handle that and how you can connect the metadata table to that. Because our data cannot be just naked, but they have to be.
Yeah, and if I can add, we should
Yeah.
try to actually do it with difficult things first,
Yeah.
right?
Yeah.
Because we really need a realistic test of how long does it take to upload,
Yeah.
does it crash,
Yeah.
data
Yeah.
corruption, all
Yeah.
of that stuff.
Yeah, absolutely. Like trying, upload a multiplex immunofluorescence, a rare site file. Let's see if he's able to handle it, uploading it and visualizing. That will be the hardest part. Because I have Vectra, you have images from rare site, I guess it's it's an Omitif or is a, Omitif, you're right. I have, maybe I don't want to try at the beginning, but I have Comet images, like they can be 300 gigabytes of images, so they're like enormous, basically. So that also you want to stress the system and see if it's able to handle all of this. So I'm aware there's a lot to this.
Yeah. And, the one other thing is not all OME TIFF images are created equal. And sometimes you have an open microscopy environment format, but the manufacturers still write the data in a slightly weird way. So that might be another layer where we, will need to filter it somehow and make sure that we convert within the Homero server to make them most useful.
Yeah, that is able to extract also the metadata from the